Chapter 17, Genesis of Cells and Complexity. The journey from simple cells to the rise of complex multicellular organisms is a captivating saga of life's evolutionary ingenuity. It showcases the milestones that transform basic biochemical systems into the intricate cellular machinery driving the diversity of life today. We do not yet know the exact sequence of steps, nor can we be certain that we have accounted for all of them. The environmental conditions on a global scale and the local conditions at the specific point on Earth where life originated remain unknown. I doubt we will ever know for sure, as the conditions then are not the same as those in today's world. Therefore, this remains speculative biology. What we do know is the start. There were no replicators or cells. And we know the end, self-replicating cells. The intermediate steps are at best educated guesses. Let us try putting things together. The blind watchmaker analogy is often used to disqualify abiogenesis, where people mistakenly assume that nature works like a watchmaker, assembling gears first and moving hands last. However, nature operates differently. It begins with the outcome, the movement of the hands, without a predefined blueprint. Consider a primitive watch with only two hands and a watchmaker as the selector. Initially, the hands move erratically, lacking direction or coordination. Precision is out of the question, but from this iteration, the basic movement of the hands becomes the minimum qualification for progression. Over time, numerous iterations occur, each with hands moving erratically. Now, the selection process favors those with some semblance of direction. As components are randomly added, those watches whose hands move in the clockwise direction are selected, while others are eliminated. With further iterations, the criteria for selection evolve. Now, it is not just the direction, but also the accuracy of the hour and minute hands moving in sync in a regulated manner. In subsequent rounds, the precision of their movements is refined, first ensuring proper coordination between the hour and minute hands, then fine-tuning accuracy to a 12-hour scale. Through countless iterations, nature slowly hones the system, selecting only those components that work effectively and discarding the rest, eventually leading to the complexity we observe today. In the analogy, the watch represents the entity, and the precision of its movement symbolizes existence, its ability to function and persist over time. Nature, as the blind watchmaker, operates through iterative processes of random variation and selection. Initially, the watch's movement is erratic, lacking stability and precision, and incapable of sustained existence. However, certain configurations emerge through random changes and environmental pressures that exhibit greater stability. Nature then selects those configurations, favoring stability and the capacity to maintain movement, ensuring that the entity has a higher likelihood of enduring and continuing its function. Each iteration improves the watch's or entity's precision, leading to a more stable and regulated existence. This process of blind selection is akin to how natural selection operates, by favoring traits that increase an organism's ability to maintain stability and survive in its environment, ensuring that those traits are passed down to future generations. The genesis of prokaryotes. The origin of cellular life is a complex and captivating journey that can be traced back to the emergence of primordial replicators in the early stages of Earth's history. Transitioning from simple molecules to protocells involves intricate molecular evolution and self-organization processes. The emergence of membranes was a pivotal event in the evolution of life, marking the transition from free-floating replicators to self-contained units capable of complex functions. Early replicators, likely RNA strands or other primitive genetic materials, existed in a prebiotic environment rich with organic molecules. However, their survival and replication depended on protection from environmental fluctuations and the ability to concentrate resources for biochemical processes. The spontaneous formation of lipid bilayers from amphipathic molecules, abundant in the prebiotic soup, provided a solution. These molecules naturally aggregated into vesicles due to their hydrophilic heads and hydrophobic tails, forming primitive membranes. Amphipathic molecules, such as phospholipids, possess both hydrophilic polar head and hydrophobic nonpolar tail regions, enabling them to assemble into bilayers in aqueous environments spontaneously. This self-assembly occurs due to the hydrophobic effect, where nonpolar tails avoid water while polar heads interact with it. This unique chemistry was pivotal in prebiotic evolution, as amphipathic molecules formed the basis of membranes that compartmentalized early biochemical reactions. Membranes provide selective permeability, protecting replicators while allowing the exchange of essential molecules. Molecules such as methane and carbon dioxide can traverse through these membranes. This compartmentalization enhanced the efficiency of reaction networks and enabled the emergence of protocells, marking a crucial step towards cellular life. Initially, these membranes may have encapsulated replicators by chance. Once trapped, the replicators benefited from a localized environment, fostering molecular interactions necessary for replication and catalysis. Membranes create selective barriers, permitting the diffusion of essential nutrients while protecting against harmful substances. Over time, these encapsulated replicators evolved mechanisms to influence membrane composition and permeability, establishing a symbiotic relationship between the genetic material and the lipid boundary. This confinement also drove competition and innovation, as replicators within membranes gained a survival advantage. The emergence of membranes safeguarded replicators and laid the foundation for the evolution of cellular life, enabling complexity and specialization to flourish. As these replicators evolved, natural selection favored the development of more stable and efficient mechanisms for self-replication. Variations and mutations in the replicator's sequences allowed for the emergence of diverse populations with varying degrees of stability. Over time, this diversity led to the selection of more robust and complex replicators, laying the foundation for the eventual transition to protocells. The dynamic interplay between genetic damage and recombination in the primordial replicator pool introduced an element of unpredictability into the evolutionary process. Primordial replicators, such as RNA molecules, were vulnerable to various environmental challenges that could cause damage. Instances of damage, such as mutations or changes in nucleotide sequences, could result in significant alterations in the functional properties of the replicators. The initial stages of life's evolution were characterized by the dynamic nature of primordial replicators, which experience genetic changes through damage and recombination. Understanding these processes is crucial for unraveling the complexities of life's origins and the subsequent emergence of mechanisms that regulate and stabilize genetic information within evolving cellular entities. The origin of cells unfolds as a dynamic interplay of molecular evolution, self-replication, and environmental influences, with cellular boundaries eventually playing a crucial role in mitigating excessive genetic change through HGT. The genesis of bacteria and archaea could have emerged from adjacent but distinct ecological niches, where early life forms encountered different energy gradients and environmental conditions. In one scenario, primitive life might have first evolved in a relatively neutral or mildly acidic environment, where simple compounds like hydrogen, methane, and carbon dioxide were prevalent. Early organisms might have relied on basic chemical reactions using hydrogen or methane as energy sources in such a setting. These organisms could have evolved into the first lineages of archaea, specialized to harness energy from these reduced compounds, forming primitive metabolic pathways. Adjacent to these environments, perhaps in slightly less extreme, more oxygen-rich or mineral-rich areas, other early life forms that could exploit different resources might have emerged. These organisms might have begun to use sunlight as an energy source, leading to the development of simple photosynthetic processes, or adapted to utilize organic compounds like simple sugars or amino acids through fermentation or respiration. These organisms would evolve into early bacteria with the ability to utilize light or more complex chemical energy sources. The most likely origin of cells in the context of bacteria and archaea would probably stem from the simpler, less complex environments where archaea-like organisms might have first emerged. Early Earth was dominated by extreme conditions, such as volcanic activity, hydrothermal vents, and anoxic high-temperature environments, where basic chemical reactions would have been more feasible. The first life forms in these environments would likely have been more chemically primitive, utilizing simple compounds like hydrogen, methane, and sulfur for energy. Archaea are well known for their ability to thrive in extreme environments, and their unique biochemistry, such as the use of isoprenoid lipids in cell membranes, suggests that they could have been among the earliest life forms, adapting to these harsh conditions. The metabolic pathways characteristic of archaea, such as methanogenesis and sulfur reduction, align with the types of chemical energy that would have been abundant in such environments. While bacteria evolved to exploit a broader range of ecological niches, including the ability to harness light energy through photosynthesis and eventually oxygen in aerobic respiration, the most primordial environments likely favored the simpler metabolic systems found in archaea-like organisms. Maybe the origin of cells in the first forms of life is most likely rooted in the archaea-like organisms that thrived in ancient extreme environments. The genesis of eukaryote. The eukaryote is a product of the archaea and the bacteria, which must have lived nearby at a point in space and time. The first of the anaerobe, archaea, could have given rise to both bacteria, oxygen utilization for combustion, and, at a later stage, photosynthetic bacteria, ability to harvest light energy. The endosymbiotic theory, explaining the origin of mitochondria and chloroplasts as ancient symbiotic relationships, was first proposed by Konstantin Mereshkovsky in 1905. Ivan Wallin expanded on it in the 1920s, and Lynn Margulis revived and significantly advanced the theory in the 1960s. Her landmark paper, On the Origin of Mitosing Cells, published in 1967, provided robust theoretical arguments and gathered molecular evidence for the bacterial ancestry of chloroplasts and mitochondria. Despite initial skepticism, her persistence helped establish the theory as a cornerstone of evolutionary biology. In the following sections, I will describe my opinions and understanding of the endosymbiotic theory and views. Two key factors likely contributed to the emergence of these novel energy transactions. I, the near exhaustion of the organic matter, and T, the abundance of anaerobic byproducts. A combination of these factors could have driven this evolutionary shift. Emergence of oxidative phosphorylation. The first prokaryotes fed on the organic matter that was naturally delivered on a daily basis, periodically, by the environment. They grew in numbers and diversified along the way. As a result, numerous prokaryotes emerged, each capable of surviving on various organic molecules. With the increasing numbers, a variant likely arose that efficiently metabolized the organic matter, extracting more from the available resources. This efficiency created a selection pressure for the emergence of aerobic respiration. Did these organisms coexist? It is plausible. One can imagine archaea and bacteria living in close proximity. This close living arrangement may have led to cooperation, where energy-rich molecules were shared. Archaea, anaerobic, could have excreted byproducts, such as lactic acid, which accumulated in abundance. Variants of bacteria might have evolved to further metabolize these energy-rich byproducts to meet their needs. There may have been matter and energy transactions between these two types of organisms. Anaerobes might have provided molecules that aerobic bacteria utilized, potentially returning ATP in exchange. Living in close quarters could have also facilitated the development of membranes encasing them together. There is a vast difference between anaerobic and aerobic respiration in terms of efficiency. Only 2 ATP molecules are produced per glucose molecule through anaerobic respiration, while 36 are produced through aerobic respiration. Anaerobes had the advantage of number as the first evolved cells, while the aerobes had the advantage of high efficiency. How did their association become irreversible? What happened to the genomes of these two separate organisms? Each was a distinct entity with a separate identity. Could there have been fusions or recombinations between their genomes? These possibilities open intriguing questions about the evolutionary dynamics that shaped early life. The genesis of mitochondria. They both would have existed in a microenvironment delimited by a common membrane. The anaerobe could have developed transport systems similar to the nuclear pore complex. On the contrary, as is popularly described, the aerobe might have entered the anaerobe, with the invagination of the aerobe's membrane forming several membrane systems, including the nuclear membrane. Regardless of how it occurred, they became closely associated. However, how did this relationship become irreversible? That is, how did two different identities become one identity? The aerobe, no longer exposed to environmental stringencies, might have experienced selective pressure to reduce its genome, shedding genes that were no longer required. Imagine a large anaerobe infested with multiple aerobes in a symbiotic relationship. One of the aerobes could have fused with the nucleus, bringing both genomes into close proximity and increasing the likelihood of recombination. Over time, parts of the aerobe's genome might have integrated into the anaerobe's genome. What were the consequences of this for the aerobes? Intense competition likely arose among the aerobes, with those that replicated and divided more efficiently being retained while others were outnumbered and went extinct. Which aerobes would have fared better, the ones with larger genomes or those with smaller ones? The larger aerobes, burdened by slower replication, higher energy demands, and unnecessary metabolic functions, would have been disadvantaged. The smaller aerobes, with streamlined genomes, would have had distinct advantages. In this context, where some aerobe genes were encoded by the nucleus, the anaerobe's genome, the aerobes that lost these genes gained a competitive edge over others. The anaerobe acquired many genes through several such events, while the aerobes that lost those genes had a competitive advantage over other aerobes. Consequently, neither of these entities, the anaerobe and aerobe, could survive independently. With its reduced genome, the aerobe lacked the capacity to replicate and sustain itself in the environment. Meanwhile, the anaerobe would not have had the efficiency of metabolism that the aerobe provided. What started as two distinct entities with separate identities ultimately merged into one entity with a new identity, the eukaryote with mitochondria. Emergence of chloroplast. The evolution of photosynthesis likely began with the development of carbon fixation pathways. Early organisms utilized chemical energy from inorganic molecules to fix carbon, as seen in modern chemosynthetic organisms. Carbon fixation allowed these organisms to incorporate CO2 into organic compounds, crucial for survival in environments devoid of light, such as deep-sea vents. Once carbon fixation was established, the need for a more efficient energy source led to the evolution of light-harnessing mechanisms. Primitive photosynthesis evolved, using light to provide the energy required for carbon fixation. This process, initially simple, eventually gave rise to oxygenic photosynthesis, enabling life to harness the abundant energy of sunlight. Upon the rise of primordial eukaryotes, there must have been tremendous proliferation, leading to the exhaustion of organic matter or intensified competition. This selection pressure could have driven some prokaryotes to evolve mechanisms to harvest energy from sunlight. Especially in cases of organic matter depletion due to extensive competition, some organisms likely evolved protein systems capable of capturing solar energy. These advanced cells no longer depended on organic matter as their primary energy source. Instead, they synthesized complex molecules from simple constituents, achieving a form of self-sufficiency that supported both self-preservation and propagation. The emergence of the chloroplast likely mirrors the process described for mitochondria, involving a symbiotic relationship between an ancestral eukaryotic cell and a photosynthetic bacterium, likely a cyanobacterium. The ancestral eukaryote, already hosting mitochondria, could have engulfed a photosynthetic bacterium through phagocytosis, but instead of digesting it, a symbiotic relationship developed. The engulfed cyanobacterium, sheltered within the host, provided the eukaryote with the ability to harness light energy through photosynthesis. Over time, the relationship became obligatory. Initially, the cyanobacterium retained its genome, functioning independently within the eukaryote. However, selective pressures favored the transfer of many cyanobacterial genes to the host's nuclear genome. This gene transfer likely occurred through mechanisms such as recombination or direct integration, as the proximity of the cyanobacterium to the eukaryotic nucleus increased the likelihood of genetic exchange. The loss of unnecessary genes in the cyanobacterium led to the emergence of a streamlined organelle dependent on nuclear-encoded proteins for its functioning. Simultaneously, competition among cyanobacterial symbionts within the eukaryote favored those with smaller genomes and faster replication. The result was the irreversible transformation of the cyanobacterium into the chloroplast, a specialized organelle. This mutualistic relationship allowed eukaryotes to colonize new ecological niches and exploit sunlight as a source of energy, giving rise to the lineage of photosynthetic eukaryotes. Most of the diverse life forms are fueled by the energy the photosynthetic organisms harvest from the sun. They change the chemistry of the atmosphere and the biology of the earth. The genesis of multicellularity. The genesis of multicellularity represents a transformative chapter in life's history, marked by the emergence of complex organisms composed of specialized cells working cooperatively for survival and reproduction. This evolutionary leap occurred independently in various lineages, including plants, fungi, and animals. Genetic innovations, environmental pressures, and ecological interactions shaped it. The transition likely began with unicellular organisms forming simple aggregations, where cooperation improved collective fitness. Cell adhesion, communication, and differentiation mechanisms emerged, laying the foundation for more intricate multicellular structures. Regulatory networks evolved to coordinate cell division, differentiation, and behavior, allowing cells to work in concert within a single organism. Intercellular connections like gap junctions and adherence junctions facilitated communication, resource sharing, and tissue integrity. Environmental factors such as predation and competition likely favored organisms with greater size and complexity. Multicellular structures provided several advantages: reduced surface area of exposure, improved nutrient sharing, efficient energy utilization, and protection for internal cells from predators and environmental stressors. This structural cooperation enabled coordinated responses to external stimuli and allowed the organism to better adapt to environmental challenges. Division of labor among cells became a defining feature, with specialized cells enhancing the organism's efficiency and adaptability. Over time, multicellularity laid the groundwork for the astounding diversity and complexity of life forms observed today. The genesis of groups. Dionysius states that Spartans excuse without penalty the warrior who loses his helmet or breastplate in battle, but punish with loss of all citizenship rights the man who discards his shield, since the helmet and breastplate are carried for one's own protection, while the shield was carried for the safety of the whole line. The environment is a constant force seeking to destabilize or extinguish life. For any organism to persist, it must possess the capacity to resist these forces or derive the necessary resources to sustain itself. On an individual level, survival requires immense effort, as each organism must fend for itself in a harsh, unyielding world. However, when organisms work together as a group, the burden of survival is distributed, leading to reduced costs and increased benefits. This is the genesis of cooperation, where the struggle for existence transforms from an individual endeavor into a collective pursuit. The evolution of grouping and cooperation allowed organisms to share resources, defend against threats, and increase their chances of survival. Group dynamics enhanced the probability of existence and improved its efficiency. As individuals began to interact and cooperate, instincts for social bonding and altruism were selected. Behaviors that promoted mutual support, such as sharing food, defending the group, or caring for offspring, provided greater reproductive success. Those individuals who participated in altruistic behaviors were more likely to thrive and pass on their genes, fostering the evolution of cooperative structures. This foundational shift towards social living formed the basis of complex communities and is a key driver of evolution in many species today. The emergence level reaches a new dynamic, with more eyes and arms to work to increase the success rate, thereby feeding the empty tummies.